Welcome, everyone. My name is Renee Duprell, and I'm the Director of Development here at the Institute for Systems Biology. Thank you all for joining us for our almost monthly Research Roundtable. This is our virtual series that we host to keep you up to date on research at ISB. This series is geared toward non-scientists, and our goal is to make our work more approachable to all. Our intention is to have one of these just about every month, so keep an eye out for our emails. Today, we'll be hearing from our featured scientists. Then there will be an opportunity for questions and answers after that. Today's topic, the impact and outcomes of COVID during pregnancy. There's a Q&A box at the bottom of your screen, and you're welcome to submit questions at any time. I'll be monitoring those, and at the end of the presentations, we'll move to the question and answer session, and we'll work our way through those questions. We'll hear from a number of our researchers who have pre-recorded their comments. Then we'll be joined by Doctors Hadlock and Piekos, who will answer your questions. Now I'd like to introduce another member of the ISB family, Carol Ellison. Carol serves on ISB's foundation board, which helps ISB raise philanthropic funding. Carol has been a volunteer and supporter of ISB for many years. She is a generous sponsor of 2 postdoctoral research fellows in Lee Hood's lab, known as the Ellison Fellows, one of whom is sharing her research with us today. Carol is an author and a poet. She's published a dozen books filled with inspiring poems. Welcome, Carol.
Thanks, Renee. I've been a supporter of ISB for many years, and I'm always amazed at the research and discoveries that come from the Institute. It gives me great pleasure to sponsor 2 postdoctoral fellows, one of whom you will hear from today. Since getting involved with ISB a few years ago, my world has expanded exponentially. I've always been curious about how things work, and ISB has more than helped to keep my curiosity gene, if you will, alive. I've also formed some wonderful friendships with some of the scientists and staff that I will always treasure. Jen Hadlock is an assistant professor and director of medical data science at ISB. Her research aims to understand the transitions between wellness and disease. In addition to studying maternal fetal health, her lab studies immune-mediated inflammatory diseases and chronic multimorbidities. Samantha Piekos is the K. Carol Ellison Fellow in Bioinformatics in the Hood Price Lab here at ISB. Upon completion of her PhD at Stanford, she joined ISB in 2020. She is studying a number of issues around pregnancy and COVID, such as understanding the impact of the COVID-19 vaccination on maternal fetal outcomes. A very warm welcome to all of our speakers today.
Thank you for joining us today for our roundtable on pregnancy research. Pregnancy research is essential because we care about mothers and babies. And most pregnancies go smoothly, but there can be challenges. The rate of preterm birth remains around 10%, and there's a need for better ways to predict and prevent this condition. And pregnancy-related mortality, after dropping for several decades, has now been rising again to 17 maternal deaths per 100,000 live births. Pregnancy is also common. In the US, about 9% of women of childbearing age are pregnant each year. However, pregnant women are often excluded from research studies. And this is partly driven by the need to protect vulnerable populations. But the end result is that medical research often lags behind, and we've seen this again with COVID-19. Researchers are encouraged to include pregnant people unless there's a good reason not to. However, they're still routinely excluded. And one of the biggest reasons is that it's hard because pregnancy adds a lot of variables and changes over time. The cardiovascular, respiratory, gastrointestinal, endocrine, musculoskeletal, immune systems all change. And those physiological changes can alter the pharmacokinetic properties of many medications. So the impact of a drug or a procedure or virus on mother and baby may be different early in pregnancy than later on. Fortunately, systems biology is all about personalized medicine and transitions over time. So we already have tools and scientific methods that we can apply to pregnancy. And conversely, what we need to do to learn how to study pregnancy well can help apply to all of systems biology because all of us are changing over time. It's also a rare opportunity to learn about some fundamentals of biology. For example, pregnancy can help shed light on understanding diseases. Some autoimmune diseases get better during pregnancy and others get worse, which gives us some clues into the nature of the mechanisms of those diseases. Today, we'll hear from 4 researchers who have been collaborating on pregnancy research, and we'll do a deep dive on COVID-19. Dr. Priyanka Balan is a senior research scientist at ISB who has been looking at multi-omic data to investigate metabolomics in pregnancy. Yami Huang is a graduate student at the University of Washington and ISB who's been working with hundreds of thousands of electronic records to look at the impact of medication timing during pregnancy. Dr. Tanya Sorensen is a maternal fetal medicine specialist at Swedish Providence, and she's been collaborating on COVID-19 maternity research and bringing her perspective from daily patient care during the pandemic. And Dr. Samantha Piekos has been looking at the impact of COVID-19 as well as real-world vaccine effectiveness. So thank you for joining us today and for supporting research at ISB.
Hello, everyone. I am Priyanka Balan, a senior research scientist at the Institute for Systems Biology. Today, I'll be talking about the impact on metabolism due to infection during pregnancy. I have been studying how metabolic changes impact various health-related outcomes such as cancer, neurodegeneration, infection, as well as pregnancy. At ISB, we have led several studies that have looked into the impact of SARS-CoV-2 infection and its influence on the immune system as well as metabolism. One of the areas of focus in our group is predicting pregnancy outcomes using multi-omics data. During pregnancy, the immune cell interactions at the maternal and fetal interface are crucial for pregnancy tolerance and labor. There's a need for research to understand better how the maternal-fetal interface operates during normal pregnancy and the pregnancy complications. Our body's mechanism to regulate energy and metabolism is closely linked with the immune response, and an imbalance in these physiological processes can lead to adverse outcomes. So it's essential to focus on the metabolic changes during pregnancy, as the metabolic signatures can predict adverse pregnancy outcomes. To identify these metabolic signatures, we have collaborated with researchers from the University of Florida, Magee Women's Research Institute, and the Seattle Children's Research Institute, and studied the placenta collected after delivery. The placenta is an essential organ for successful pregnancy, as it performs critical functions such as transferring nutrients and oxygen to the fetus, removing harmful waste products, and providing mechanical, hormonal, and immune support to the developing fetus. Alterations to the placental function can have profound impact on the health of the developing fetus and pregnancy outcome. Using the placental samples, we have carried out analysis of how genes are expressed, which metabolites and proteins are present in abundance, and how this information can be used for generating computational models. Our goal here is to develop a predictive model that combines data from various omics measurement and can be used to predict the metabolic trajectory across pregnancy. So combining this information on metabolic changes with the ongoing studies on COVID-19 infection in pregnant women will be beneficial. It will allow us to explore how viral transmission and infection affects the fetus during pregnancy. In recently published studies, the researchers observed increased incidences of gestational diabetes and hypercoagulation in pregnant women with COVID-19 infection. Also, studies have shown an increase in the levels of ketone bodies in the urine of pregnant women, suggesting a perturbation in glucose and lipid metabolism. Thus, we need to pay attention to the high risk of metabolic complications such as ketonuria, hypercoagulation, and gestational diabetes in pregnant women with COVID-19 during late pregnancy. With the spread of coronavirus, pregnant women and their fetuses have been concerned as a high-risk population. Some studies have reported that the placentas have pathological changes and abnormal weight when the mothers were infected with the SARS-CoV-2 virus during late pregnancy. The interactions between coronavirus infections and pregnancy is worthy of study to identify immune and metabolic markers that have clinical significance. Thank you.
Hi, my name is Yami Huang, and I am a PhD candidate at University of Washington Molecular Engineering Program. I currently belong to Hadlock Lab, and my main focus of research has been on detecting and validating drug effects during pregnancy using AHR data. As you may be aware, drug effects on pregnant women are under study. Randomized clinical trials rarely include pregnant women unless the product is targeted for pregnant women. Therefore, efficacy, safety, and dosage all are determined based on men and non-pregnant women. In fact, 97.7% of newly approved drugs from 2000 to 2010 had insufficient data to assess the impact on babies, and 73% of them had no data to determine the safety in pregnancy. Despite inadequate pregnancy safety information, most pregnant women take medication. Overall, 94% of pregnant women take at least one medication, over-the-counter or prescribed medication combined. And in terms of prescribed medication, 49% of pregnant women take medication in the US, and this ranges from 23 to 96%, depending on the country. We believe electronic health record is a promising data source to study effect on pregnant women, as it contains rich data on longitudinal phenotype, confounders, and prescription orders. As mentioned earlier, my main focus of research has been on detecting and validating drug effects during pregnancy using AHR data. To detect the previously known and unknown signals, I have been using both statistical data mining and machine learning method. And in order to validate new signals or previously known to be controversial signals, I apply traditional pharmacoepidemiology method. Pharmacoepidemiology is a study that bridges between pharmacology and epidemiology. As a part of the effort, I have looked at the timing effect of the timing of SSRI, which is a major class of antidepressant on pregnant women. And for this study, we limit our study population to women who had depression before the pregnancy in order to avoid any confounding by depression itself. We also assess the impact of the prenatal and pre-pregnancy depression severity using the standardized depression severity test score. Such depression severity score is difficult to collect for a usual or normal retrospective study. However, this could be collected because our data set is AHR data and was part of a prenatal routine care. As an ongoing effort, we will have been looking into immune-mediated inflammatory disease and drugs for those diseases among pregnant women, and also looking into anticoagulant use during COVID-19 infections among pregnant women. We will keep you updated with future results, and so please stay tuned for upcoming results. Thank you.
So I'm Tanya Sorensen. I'm a maternal fetal medicine physician at Providence Swedish at First Health. And I have been working with this Institute for Systems Biology for quite a while. I've been on their scientific advisory council, but I've been really, really fortunate to work with Dr. Hadlock more recently on some COVID research. It's hard to describe what it was like when COVID came to us. It sort of showed up in the US here in the Northwest region, and we had really no idea what to do with it. We knew that things were terrible in China, and we had zero idea of how COVID was going to affect pregnant women. And in fact, a lot of viruses can be very hard on pregnant women. And so we were looking at what, if it was going to be like a bad flu, we really didn't know. And at the same time, if you think back, there were problems with PPE. There was a lot of fear. There was a lot of uncertainty. And so it has just been a huge honor to be able to work with ISB and Dr. Hadlock's team to be able to shed some light on what COVID is in pregnancy and how it affects pregnant women. I was just telling Dr. Hadlock that I didn't realize I was clinical today. And so that's why I'm actually sitting in my call room on my phone talking to you. But we actually have a COVID pneumonia patient in the hospital again, which hasn't happened for a really long time. As it turned out, COVID in the beginning was not as hard on pregnant women as we thought it might be. But by the time the Delta wave came around, we had multiple patients in the ICU and we had maternal deaths from COVID. This was pre-vaccine. And we were desperately pleading with women to get vaccinated. And of course, it's a scary thing the COVID vaccine is a fairly new technology, and it's a scary thing for pregnant women to make decisions about new technologies when they're, carrying a baby and worried about what might happen. So we've been partnering, and I have contributed sort of a clinical sort of lens on the work that Dr. Hadlock's been doing, and have been able to just advise a little bit on, what are the questions that we want answered? What are the best ways to look at that? What's going to be really useful? And so really what's come out of this has been so much. With Providence, we have a very large database. I think there are around 70,000 deliveries a year within the Providence system. And so, and of course, where I am is the quaternary center. So we see the sickest patients here. And so we have a really big database. And so we were able to really drill down on, what happens with COVID and pregnancy. And we're able to actually find some real results that others hadn't really had a chance to come up with yet. So we do know that COVID can affect pregnancy, it can affect fetuses, it can be, especially certain variants have been very dangerous for pregnant women. And then more recently, there's been some work around the vaccine and what happens with vaccinated patients that's been really promising. So so really, I guess that my message is that we're so fortunate to have this team of scientists to partner with us clinicians to really come up with some meaningful information that really will inform, going forward on an incredibly tricky disease. I've been saying lately, if I wasn't so exhausted by it and it wasn't so hard, it would be really fascinating to be a doctor in a pandemic, because we don't see that. We see that every 100 years. And ISB has kind of helped me keep that view a little bit, because we really have been able to produce some interesting and really helpful information. As far as how the information that we have has changed our practice, we were not able to do this as a prospective study. We had to basically go back and look at our patients. The advantage that we had was we had so many patients and we're able to capture them through the big data that ISB is so famous for. And so we were able to come up with information quite quickly compared to other places. I have to say, in the beginning of the pandemic, so for example, when the wave hit New York really hard, there was information coming out of there, but it was very anecdotal, meaning it was information that people were just saying, well, this is what we saw, but it wasn't very scientific. And so the scientific papers have been slow in coming, and we're, I think, among the first to get some really good information out there. So what I say in the beginning, it really informed us? No, but now I can clearly tell a patient that, COVID in the second trimester of pregnancy has the potential to affect her fetus. I know when she gets COVID, I need to follow that pregnancy more closely. I need to look for signs of preterm delivery, of signs of growth restriction. And honestly, we've found an increased risk of stillbirth as well. So that has helped us a lot more recently since we've gotten that information. And I think we'll be able to also say unequivocally that vaccination is the right thing to do for pregnancy, and it actually reduces all those complications pretty dramatically. So I would say clinically that this information is very, very applicable. It's very useful. It's not dry kind of science. It's like really on the ground, informing how we can talk to patients and what we do.
All right. So thank you to all the speakers that came before. I am Sam Piekos. I am a postdoc with the Hood Price Group at ISB. And today I am going to go over some interesting results we've had on looking at COVID-19 during pregnancy. As you've heard from the previous speakers, it became well known very quickly that pregnant people are at increased risk for adverse outcomes during a maternal COVID-19 infection compared to non-pregnant people. So they're at increased risk for ICU admission, mechanical ventilation, and death during a COVID-19 infection. The vaccine there was a lot of vaccine hesitancy in the pregnant population at the beginning, which was understanding given that pregnant people were excluded from all clinical trials for COVID-19 vaccines. But what we could see is over time, the vaccination rate has gone up for pregnant people. And as of May 7th, 71% of all pregnant people in the US were vaccinated. So as you've heard before, we are partnered with the Providence St. Joseph healthcare system. So this gives us access to EHR data from over 50 hospitals and 1,000 clinics spread across 7 Western US states, which include Alaska, Washington, Montana, Oregon, California, New Mexico, and Texas. So this is really great that ISB has this partnership with Providence to give us access to this very diverse group of patients so that we could quickly respond to studying COVID-19 infection vaccination during pregnancy. And then just another little background, because I'm going to mention this throughout my talk, is we typically do perform matching of patients. So for example, we are not doing a randomized control trial for patients that got infected with COVID-19 during pregnancy or not, or patients that got vaccinated against COVID-19. So there are differences in the composition of the populations of people that had these outcomes. And with that, if there's a lot of underlying factors that might be involved given the composition of these cohorts. So in order to control for these other factors such as type of insurance status, do you smoke, that sort of information, demographic, lifestyle, clinical comorbidities like chronic hypertension. We use machine learning to match. So we want to make sure that we are comparing like with like for patients of these 2 different populations, because what we want to be confident in is the signal that we're seeing is from the COVID-19 infection or COVID-19 vaccination and not an underlying difference in the cohorts. So just an overview of what I'm going to be talking about for the next 15 minutes. We're going to go over the impact of COVID-19 infection on birth outcomes and then go into COVID-19 vaccination, effect on maternal fetal outcomes. But first, let's start with COVID-19 infection during pregnancy. So what we're looking at here is pregnant people that delivered either at term or prematurely following a COVID-19 infection during the first trimester in pink, the second trimester in blue, the third trimester in green, and then the gold is this matched negative control. So that's why I was just mentioning earlier, we matched our patients that didn't get COVID-19 during pregnancy with those that did, so that they had a similar composition. So that's what we're comparing to in the gold, is that matched control. And what we're seeing is overall, patients that had COVID-19 during pregnancy had an increased risk for preterm birth. And what we're seeing here is there's a trend that the highest rate of preterm birth was during the first trimester infections during pregnancy. Another thing that we evaluated is small for gestational age. What this is it's birth weight adjusted for gestational age at delivery. plus fetal sex. And so what this is looking for is the baby smaller at delivery than expected based off of gestational age. So we're looking at the bottom 10% of the fetal growth percentile for this. And what we're seeing is overall, if a pregnant person had COVID-19 during pregnancy, they were significantly more likely to have a baby that was small for gestational age at delivery. And what we're saying is this is largely driven by a third trimester infection. That's where we're seeing the biggest increase. We also considered other outcomes like stillbirth. So going through here, what we looked at the subtotals for each trimester infection, as well as all of them together. We saw 7 stillbirths in our SARS-CoV-2 positive cohort, whereas in our matched negative control, we only saw 1. So this is what we would expect from the population would be 1. We saw 7. So people that had a COVID-19 infection during pregnancy were at increased risk for stillbirth. And then we're also seeing during the first and second trimester infection, those babies tended to be born at lower gestational age. And then birth weight was lower following a first and the third trimester infection. Now, the cool thing about having access to all of this data is that we can use big data techniques like machine learning to build models to project things like gestational age at delivery following a maternal COVID-19 infection. So we can build these models, and then we can look under the hood to see what's driving these models to make the predictions of the gestational age. So we can say, what are these models finding important to understand what is going on with the maternal COVID-19 infection that can be driving this lower gestational age at delivery. So what we're looking at here is the contribution of each individual feature. So each factor that was put into the model for predicting gestational age at delivery. At the top, we see our most important feature, which is gestational age at COVID-19 infection. So when during pregnancy a person was infected. And what we're seeing in the midline here is this is, 0. Think of this as 0. And then to the right, this would be promoting higher gestational age. To the left would be a lower gestational age at delivery. And what we're seeing is people that tended to get infected early on in pregnancy tended to have lower gestational age at delivery. And we can go through. So the cool thing about that machine learning model is it was taking into account over a dozen features all at the same time. And that is interesting to know how they interact. We can also look at each individual feature one-on-one with gestational age at delivery. So just do a correlation. And what we're seeing for gestational age at delivery versus gestational age at infection is that there is some association there. What's interesting is some patients follow that, where gestational age at delivery was closely associated with this trend line with gestational age at infection. But for a lot of patients, it didn't seem to impact their gestational age at delivery. So it seems like this is important for a subset of patients that had maternal COVID-19 infections, but it doesn't hold true for everybody that had an infection during pregnancy. But it's really important that we investigate why for these patients there's this negative outcome of this earlier gestational age at delivery. And we can do this for all the other factors we looked at, which include the number of medications a patient received during a COVID-19 infection, with higher number of medications being associated with earlier gestational age at delivery, and other things like how severe the infection was. And what was interesting here was the severity of the infection was uncorrelated with gestational age at delivery. And same with the number of medical encounters related to a COVID-19 infection. There was no correlation with that with gestational age at delivery. So what this is suggesting is that there's an increased risk for just earlier gestational age at delivery, regardless of COVID-19 severity. So what I just went over is that maternal COVID-19 infection increases the risk of negative birth outcomes, including preterm birth, stillbirth, small for gestational age, and decreased birth weight. The biggest predictor of gestational age at delivery is gestational age at infection, and there is no correlation between the severity of the disease and the adverse outcomes. So regardless of disease severity, there's an increased risk for negative birth outcomes. And if a person is pregnant and has a COVID-19 infection or suspects of having a COVID-19 infection, they should definitely reach out to their doctor and share that information. All right, now going on into COVID-19 vaccination impact on maternal fetal outcomes. So this was great when the vaccine came out because this allows a preventative approach to help mitigate the risk for COVID-19 infection. And so just a few brief definitions before we get into the meat of this. Unvaccinated, when I use this term, I'm referring to somebody that has no record of COVID-19 vaccination, nor have they had a COVID-19 infection recorded prior to the start of their pregnancy. An mRNA vaccine, this is referring to specifically Moderna or Pfizer COVID-19 vaccine. Full vaccination status, so I'm referring to the 2 weeks after the second shot of the mRNA vaccine is when somebody is considered to have the full protection of the vaccine. The booster is a third shot that was given more than 5 months after the last dose of the primary series. So after the 2 shots, you get a third dose after 5 months. That was the booster. And then a COVID-19 infection was a positive PCR COVID-19 test that's recorded in the EHR. So let's just look at over time, the number of people that were vaccinated at delivery. So what we see is going from the end of January of 2021 through May 4th of 2022, we see a decrease of the unvaccinated individuals over time, mirrored by an increase in people that are vaccinated or boosted here in teal. We also looked at the number of infections that were contracted during pregnancy. So if we look at the unvaccinated in red, we're seeing a larger number of infections. Over here, this bump would have been during the Delta wave. So those were primarily driven by unvaccinated people. We didn't see very many infections in the vaccinated individuals. When we got to Omicron wave right here, we saw a huge spike in the unvaccinated individuals. And we did see some breakthrough infections in the vaccinated as well, but not really in the boosted population here in teal. If we, on the left was raw counts, on the right, what I have is the percentage of people at delivery that had a COVID-19 infection at some point during pregnancy. And what we're seeing is unvaccinated people were significantly more likely to contract COVID-19 during their pregnancy than vaccinated or boosted, and that boosted people had an even lower percentage of people that had COVID-19 during pregnancy compared to people that just received 2 doses of the vaccine. And what's important to note is that this has gotten incredibly common. So some weeks we're reporting over 30% of patients that delivered in the unvaccinated cohort had a COVID-19 infection during pregnancy. And looking at patients that did have that maternal COVID-19 infection, we asked what level of care they received during that infection. And so on the left, we have our vaccinated population. On the right is the unvaccinated population. And what we're seeing here is a decrease in emergency care here in purple needed by people in the vaccinated population compared to unvaccinated population. So the vaccinated people that had a maternal COVID-19 infection were significantly less likely to need emergency care during that infection. Now, looking at a broader range of maternal outcomes from a COVID-19 infection, we have the vaccinated in green, the unvaccinated match. So this is what I talked about previously with controlling for underlying factors in the composition of the cohort in orange, and then the unvaccinated in red. Looking at the percentage of people with a COVID-19 infection during pregnancy, their vaccinated people are significantly less likely to get COVID-19 even after, even compared to the matched cohort. Looking at hospitalizations, vaccinated people were significantly less likely to be hospitalized compared to unvaccinated people, but there was no difference compared to the unvaccinated matched cohort. Going into the percentage of people that received vasopressors during a COVID-19 infection, vasopressors are a type of medication to help stabilize a patient's blood pressure. So this is usually administered when a patient's blood pressure goes dangerously low. We're seeing no difference in the usage of vasopressors during maternal COVID-19 infection between the vaccinated and unvaccinated population, but if we compare to the unvaccinated matched, we do see a significantly less people in the vaccinated cohort receive vasopressors. And then across all three populations, we saw no difference in supplemental oxygen use during a COVID-19 infection. Now, that's the outcomes for mom during the acute COVID-19 infection. What does this, what about the impact of vaccination on birth outcomes? So looking at all people that were vaccinated, looking at birth outcomes that occurred after the vaccination, we have, again, the vaccinated in green, the unvaccinated matched in orange, and then the unvaccinated in red. And looking at preterm birth, vaccinated people are significantly less likely to have a baby that's born prematurely. They are less likely to have a stillbirth and less likely to have a baby born with very low birth weight. So this is less than 1,500 grams during at delivery. And this is true when comparing the vaccinated patients to the total number of unvaccinated patients, as well as the matched control. Looking at the distribution of the timing of the preterm births. Preterm is a broad category where a baby is born at less than 37 weeks gestation, but there's very different outcomes depending on if you're late preterm versus if you're extremely preterm. So late preterm babies tend to do very, very well, whereas extreme and very preterm tend to have extended stay in the NICU and higher mortality after delivery. So what we're seeing here is that for babies that were born prematurely to the vaccinated individuals, they were significantly more likely to be late preterm and less likely to be very or extremely preterm, like we're seeing in the unmatched and unvaccinated matched and unvaccinated cohorts. So that's important because the longer we can delay delivery for these babies, it makes a lifelong impact on their health. Boosted individuals also had better outcomes than those that received only 2 doses of the vaccine. So we're looking at people that got vaccinated, got 2 shots, and then more than 5 months passed and they did not receive a booster prior to delivery. So that's in gold here. In teal, we have individuals that received 3 shots, so 3 Moderna or Pfizer vaccine shots. And what we're seeing is that there's people that are boosted had lower rates of maternal COVID-19 infection. They had lower rates of preterm birth and lower rates of low birth weight. So again, we're seeing better maternal fetal outcomes for people that received 3 shots compared to those that only received 2. So what I showed you here is that vaccinated people are significantly less likely to have a maternal COVID-19 infection during pregnancy. Babies born to vaccinated people are less likely to be preterm, stillbirth, or very low birth weight compared to unvaccinated people. Boosted people are less likely to have a maternal COVID-19 infection, deliver prematurely, or have a low birth weight baby than vaccinated but not boosted. And altogether, this suggests that this is very reassuring information about the COVID-19 vaccination during pregnancy. This supports the recommendation that pregnant people be vaccinated. And not only that, they should stay current with their vaccination series. So what does this mean going forward? Well, the cool thing about working with ISB and with big data in this project is we built a lot of stepping stones that will, and building blocks that can be used to address future problems. So I went over to how we're using big data and machine learning, as well as EHR data to give us insights. We can use all of these tools to apply to other problems as well, such as other perturbations during pregnancy or questions about drugs taken during pregnancy, any sort of outcomes, or we can even get into things like environmental exposure and look at how that impacts the individual's health. So this is what we can do moving forward. All of these are building blocks where we can use this to address other questions about what's going on with pregnancy. And overall, going forwards, our goal at ISB is to realize precision medicine. And so pregnancy is really cool because what goes on during pregnancy has a lifelong impact on an individual's health. And so the idea is we want to take all of these different types of data like EHR combined with other biological metrics and wearable information and try to understand how does that affect, how can that be used to interpret what's going on with the individual's health and how we can use that to predict and prevent adverse health outcomes. And so that's a wide-ranging goal of a lot of groups at ISD is to realize this precision medicine, this personalized medicine. I would like to take the time to acknowledge my amazing students. So part of being at a nonprofit research institutes, we train a lot of students. So I want to recognize Suhana Betty, Anissa Holloman, Koa Wong, and Padma Gudanapani for their efforts and contribution to the work I presented today. I would also like to acknowledge my colleagues as well. Jennifer Hadlock, Nathan Price, Lee Hood, Yami Huang, Ryan Roper, and Tanya Sorensen for all of their work with helping realize this project. I would like to thank funding NICHD as well as Carol Ellison for supporting my postdoc, my work, and of course, Providence for the data. And thank you for your attention. And at this time, we're going to turn it over to questions.
Yeah, and I think, thank you, Sam. That was great. I think Jen Hadlock is here. She's going to join us. So just as a reminder, if you want to pose a question, please go to that Q&A box at the very bottom of your screen and just type in your question there and we'll start working our way through those. We do have some questions. I'll kick us off. What advice can we give to women who are planning to conceive during this pandemic time? What precautions can be taken to prevent adverse pregnancy outcomes?
The number one thing that you can do is get vaccinated, get boosted. So the COVID-19 vaccination, as we just showed, has better maternal fetal health outcomes. And it's really, it helps prevent that maternal COVID-19 infection, which is important because that we really want to avoid pregnant people contracting COVID-19, the increased risk for mom and baby. Great way to do that is to get vaccinated.
Okay, great. This is an interesting kind of, oh, sorry, go ahead, Jen.
Oh, I just wanted to add as well that something that may not be obvious is that you can talk to your doctor before conception as well. And so if you're planning to get pregnant, you and the donor, father, whoever else is involved in the process, whole family, can come and talk about your personal health, your family history, your goals, and they can talk about tests that might be interesting that would help provide information, which medications you might want to start or stop, and a range of other useful information about pregnancy.
Super. And this is actually kind of this next question kind of follows up on this. Right now, I think a second booster is only recommended for people over 50 and 4 months after the last booster, but should pregnant people also be considered as good candidates for a second booster?
I'll take that one too.
Okay.
So we're really excited about Samantha's research, which suggests the answer seems to be yes, from what we're seeing. However, when deciding overall policy and recommendations, there's usually a process where committees of experts look at all of the evidence from this study and others before making decisions about that recommendation. So our evidence suggests the answer may be yes, but we aren't in a position to be able to make a medical recommendation, but your doctor can, and you could take this evidence, and because the pandemic is so fast moving, they can weigh that together with everything else they know and think about your personal situation.
Okay. Great, thank you. Next question. What implications does this have for other infectious diseases that pregnant people might contract during pregnancy?
Yeah, that's a great,
Good one.
that's a great question. So there's been a lot of work on this for the flu in particular. We do know that infections during pregnancy does raise like the risk for adverse outcomes. So that what we found with COVID-19 is consistent with other infections. So again, like this just contributes to the knowledge of what does it mean for a pregnant person to be infected during pregnancy. How is that interfering with the course of a normal, healthy pregnancy? This all can be put into that broader context.
This is actually one of the things that's been exciting. Is certainly COVID itself is very important, but the immune system changes during pregnancy, actually profoundly, where it is decreased in some ways, and yet people are usually able to fight off, infections and colds and flus throughout pregnancy. And so, understanding more about when and how infection is concerning, when and how vaccination is beneficial and appropriate during pregnancy is very interesting. And if we could get into some of the underlying fundamentals of biology, that might lead to more personalized suggestions and potentially new ways to prevent or treat these diseases. And there, the work that Priyanka and Samantha are working on with and Yami with omics is going to provide information that we can't get, even though it is on fewer people. It'll be much more breadth and depth of multi-omic information to help shed different insights.
Yeah. Can you talk a little bit more about, we heard from Dr. Tanya Sorensen at Swedish, and she's actually on the front lines treating pregnant women. And it's so exciting that our research made it over there so quickly. I mean, we worked with them, but it's great that she's actually applying it. Can you talk, what does that look like kind of on a day-to-day basis when you're working with a doctor like that? How does that work between ISB and Swedish Providence?
So it's very interesting because it's really compelling. It's a big part of our lab is this opportunity to work with Providence. And we do it in three ways. On a daily basis, we're working with the data. And the reason that's interesting is that size of patients, that collection of 200 million different records that we can look at that are updated daily, allows us to answer questions about substratification, about very specific different types of patient situations that would be very difficult to do. A lot of academic healthcare centers where most research is done, they don't have that large of a population, and it's not as general of a typical representative community population. So it really is a unique opportunity. And apart from looking at insurance claims, which don't really have the depth of medical records, you rarely get those numbers of that size. Then the second part is being able to just reach out anytime and ask an expert. So I have very general medical knowledge, but people in specialties have done 3 to 7 years of additional training in their specialty, and then have seen a physician over their life will see thousands to tens of thousands of patients and patient situations, and see them over time as they change in their life. So that is very helpful to be able to reach out. But the thing I think is most important is prioritization. So we can ask Tanya, what are the things that are most problematic for you and your patients? Where is it that decisions are difficult and more research or more information would help right now today? So risk prediction models, we can actually make a difference immediately. Or what are questions that are profoundly challenging that need to be investigated longer term? So that bedside insight and overall expertise allows us to do work that would just be impossible on our own.
Yeah, it was fun to hear from her. And so kind of a follow-up too, because you mentioned the millions of data records, because can you talk about that? Are there names attached to those? Do you know who those people are?
Having worked with patients, having been a patient, right, each and every individual, we understand that sensitivity of that data, the personalized nature of that data. And there's one of the reasons that this research is not often done is that there were actually years of work put in order to make sure that we have all of the training, technical safeguards, ethical, legal, appropriate, respectful safeguards in to respect this data. It's a really rare opportunity, and we're very grateful that we're able to use it. So that's a big part of the time that it takes. Now that we've established that, we can jump on things like COVID. We're able to look at that very quickly. But if you're starting from scratch, it is a multiple year process to set it up.
Right. And yeah, we're very fortunate and thrilled to have this partnership with Providence. So I think, let's see, we're at 4:55. So I think it's time to wrap this up. Thank you both for joining us, and for everyone else who did too. It was great to see a lot of faces, and it was really great to hear from our partners on the front lines too. So we'll be sure to thank Dr. Sorensen for jumping in with us. So everyone else, thank you for joining us tonight too. Our next research roundtable is on June 7th. And the topic is going to be DNA sequencing in the high school classroom. So we're going to hear from one of our researchers here at ISB, and we're going to hear from a teacher at Lincoln High School. So that'll be really great. That's June 7th. So if you're on our email list, you'll get notifications about that, and please do sign up. If you are inspired to make a gift, you can go to isbscience.org and click on our donate button, and we'll be thrilled to take that from you too. So thank you so much, and hopefully we'll see you next month. Have a lovely evening.